Hello everyone. This chapter will focus on the cell. What are the learning outcomes? There are 5 learning outcomes in this chapter. First, we are going to have a brief introduction on the cell. Then we are going to dive in cell structure and function. After that, we are going to look into cell division and death. Finally, we're going to take a look at stem cells and the microbiome. Now, what are cells? Cells are defined as the basic units of life. What this means is that in your body, cells are the smallest component of your body that have all the characteristics of life. They are able to reproduce, they are able to respond to their environment, they are able to use energy, and they are able to evolve, if you will. Now, in the body, there are more than 290 specialized cells or differentiated cell types. These cells aggregate together based on their similarities, forming tissues. In the body, there are 4 major kinds of tissues that occur in your body. 1. Epithelial tissues. 2. Connective tissues. 3. Nervous tissue. And 4. Muscle tissues. The tissues are characterized by the fact that they are going to share a number of features that are going to be common. Each tissue has specialized cells, and these cells are going to be different one from another. Say, for instance, nerve cells are going to be different from muscle cells because of the difference in the sets of genes each specialized cell is expressing. So here we have a summary table of the different tissues. We have the connective tissues. What is their role? The role of the connective tissue is in general going to be to surround some material, protect, provide some support, binds structures together, fill space in the body. Some examples of connective tissues are, for instance, cartilage, bone, blood, and fat. The next set of tissue we are going to address, it's epithelium. Epithelium are this tissue that are made up of cells that are tightly packed together, and they are always found lining surfaces to provide protection. Some of them, not all of them, some of them are going to secrete chemicals, some are going to absorb, and some are going to excrete substances. For instance, if you consider your digestive tract is filled with mucus. There are some cells in your digestive tract that which job is to secrete, to produce mucus. These cells are called goblet cells. The next tissue, the muscle tissue. Here, the specialized cells, muscle cells, are going to be involved in movement in the body. These cells are going to be able to contract, while the nervous tissues, which is made up of nerve cells, is going to be more specialized in transmitting information such as electric chemical impulses, the nervous tissue is going to be responsible for coordinating movements and also sense and respond to environmental stimuli. We are going to have different kind of cells in nervous tissue. We're going to have neurons and we're going to have neuroglia. Neuroglia are the cells that provide support to neurons. There are different kind of neuroglia, but I'm not going to bug you with this in this course. So, in our body, as we consider all the cells that we find in our body, there are really two major types of cells in our body based on the amount of genetic information they contain. One is the somatic cells, two is the gamete cells. The somatic cells, also called body cells, are going to be the cells such as skin cells, muscle cells, nerve cells, liver cells, brain cell. All these cells actually are going to have two copies of the genome. One copy that you receive from mom and one copy that you receive from dad. So, as human, we have 23 pairs of chromosomes. These 23 pairs of chromosomes cause the somatic cells to be called diploid cells because the genetic information is found in one copy you receive from your mom and one copy you receive from your dad. The gametes, on the other hand, are the cells that are reproductive cells. They contain only half the amount of information you would find in somatic cells. So, because of that, these cells, gamete cells or reproductive cells, are what we call haploid because they contain half the amount of genetic information. In human, we have sperm cells that are produced by males. Egg cells are produced by female. These two type of cells are the reproductive cells in human. Now, sperm cells and gametes, sperm cells and egg cells are going to come together through a process called fertilization to actually restore a diploid entity. So, a sperm cell brings 23 chromosomes, an egg cell brings 23 chromosomes, so together, now we have 46 chromosomes or 23 pairs of chromosomes. Now, in our body, we are going to have cells that are going to be responsible for the production of all other type of cells in our body. These cells are called stem cells. Stem cells are very unique in the fact that they are able to renew continuously. These are blank cells that can differentiate and produce nerve cells, blood cells, muscle cells, and so on and so forth. Right? While some stem cells, as they divide, they produce other stem cells so that there is always blank cells from which the body can go to produce more differentiated cells. As we look at the different cells that are found across living systems, really those cells in terms of structure and molecular organization fall into three categories. We have three domains. This domains is going to include the archaea, the bacteria, both of them being prokaryotic cells. And prokaryotic cells are primitive cells, very early cells that actually do not have their genetic information DNA enclosed in an envelope. Archaea and bacteria are unicellular organisms, right, that do not have a nucleus. Then the third domain is going to be the domain of eukarya. This is where we are going to find all eukaryotic organisms. Here, we're going to find some eukaryotic organisms that are unicellular, some are multicellular. Protists, for instance, are unicellular, while humans, dogs, pigs, rats are multicellular eukaryotes. So, regardless of the type of cell, whether you're talking about archaea, bacteria, or eukarya, all the cells are similar on the fact that they all have a plasma membrane, they all have a cytoplasm, which is a semi-fluid substance. They all have DNA as their genetic information, and they all have ribosomes, which is responsible for producing proteins. Eukaryotes are going to be much more complex than bacteria in the sense that bacteria, which are prokaryotes, are not going to have many compartments. So they have a plasma membrane, a cytoplasm with structures that are not really enclosed in membrane once inside of the cytoplasm, while eukaryotic cells are going to have, in addition to the plasma membrane that sets the limit of the cell, you are going to find inside many compartments. For instance, the nucleus, it's a membrane-bound organelle. The endoplasmic reticulum, both smooth and rough endoplasmic reticulum, the Golgi apparatus, mitochondria, are all organelles that are enclosed in a membrane, right? So eukaryotic cells are much more complex than prokaryotic cells that way. We as human, we are eukaryotic organisms. Therefore, our cells are relatively complex compared to bacterial cells and archaea. Now, what are the chemical components of the cell? There are four major chemical components or macromolecules. We're going to have carbohydrates. In the group of carbohydrate, we have sugars, starch. The second category of molecules of life, lipids. Lipids are, for instance, fats and oils. They don't like water, so they don't mingle well with water. Okay. The third category of molecules of life, it's going to be proteins. In the group of proteins, we're going to have some structural proteins and some functional proteins. If you consider, for instance, your hair, your nails are hard. Your hair is what it is because of the proteins that make up the hair, right? But on the other hand, you have functional proteins that are, for instance, enzymes. They are doing a specific job. They break down chemicals and so on. Finally, but not least, we're going to have nucleic acids. Nucleic acids include DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The carbohydrates in the cell are essential for energy production or are used as source of energy, they also play very much a structural role. The lipids are going to be very important for productions of hormone in our body. They are also going to be essential for the organization, the structural of organization of membranes. They can also be used as insulation and store energy. The proteins are going to have many different functions. Some of them are going to allow movement. These are contractile proteins, okay, forming fibers. Some of them are going to be essential for blood clotting. Some are found in our tissue, forming bulk structure, essential for bringing different tissues together in the connective tissues. Some proteins are enzymes. These are important in helping chemical reactions occur in our system, right? They're going to catalyze a number of reactions. Okay, they are going to be essential for the organization of the cell and the body. The nucleic acid is going to enclose the genetic information as we saw in chapter 1. That genetic information enclosed in DNA is going to be converted through the process of transcription into RNA, and RNA through the process of translation is going to allow the conversion of genes from DNA to proteins, right? Now, let's take a look at the cell components. Now, here we have a generalized animal cell. Now, this animal cell shows a number of structures, including the plasma membrane right here that sets the limit of the cell. We are going to have inside of the cell a huge amount of organelles. You can see, for instance, a mitochondrion right here. You can see the Golgi apparatus right here. You can see peroxisomes right here, lysosomes, the nucleus, a huge part, the endoplasmic reticulum right here. Okay, so many different components make up an animal cell, such as your cell. So in the next few slides, we are going to dive into the different structures that make up the cell. The first step we are going to make, it's right on defining organelles. Organelles allow a eukaryotic cell to be organized in a such way that different jobs are going to happen in very specific compartment. This allows to separate the chemical reactions and allow special compartment to play a very specific role, so there is no replication and there is more of the chemical reactions occurring in a more effective manner. So the organelles are going to keep biochemical reactions separate. Some very specific chemical reactions are going to happen in one organelle while not happening in another organelle, right? So the organelles are going to allow this organization of the functioning of the cell that is going to be optimum. Some organelles, such as mitochondria, are going to be essential in acquiring energy, while others are going to be essential in causing the destruction of foreign particles that come in, or the digestion of the particles that can be engulfed because they are a problem to the body, or organelles that are otherwise now not useful anymore. They are damaged. They need to be destroyed and be replaced. So the first step we are going to make in the different organelles is going to be looking at the nucleus. The nucleus is composed of a nuclear envelope. That nuclear envelope has little holes. These little holes are called nuclear pores. The nuclear envelope sets the limit of the nucleus, and inside of the nuclear envelope, we are going to have the nucleoplasm. Okay, the nucleoplasm, it's all this large ocean of blue stuff. Okay, so this is basically a mix of proteins, salt, and DNA that is organized in chromatin or chromosomes. Okay, so we have the nucleoplasm, and in the nucleoplasm, we are going to have DNA. That DNA is combined with proteins that are called histone. We covered that in the previous chapter. Now, notice an area right here that is very dense. That area that is dense is called the nucleolus. The nucleolus is where RNA, specifically ribosomal RNA, is produced in the nucleus. This is where the ribosome start their assembly. Nevertheless, the complete assembly of the ribosome does not occur in there. The assembly starts in there, but it's not completely done in the nucleus. So, outside of the nucleus, we are going to have an endomembrane system. But what is an endomembrane system? An endomembrane system, it's a series of membrane-bound organelles. These membrane-bound organelles somehow communicate with each other so that they can work closely with each other. So, again, outside of the nuclear envelope, we are going to have the endoplasmic reticulum. There are two kinds of endoplasmic reticulum, the smooth endoplasmic reticulum and the rough endoplasmic reticulum. We are going to address that in a minute. Then, a little further, we are going to have the Golgi apparatus that works closely with both the nuclear envelope and the endoplasmic reticulum. Then we're going to have lysosomes and vesicles. There are many kinds of vesicles in animal cells. So how does the endomembrane system function together? Now, again, you have here the nucleus, right, with the nuclear envelope, and notice how the nuclear envelope communicates with this network of sacs here. That is the rough endoplasmic reticulum. And notice that the rough endoplasmic reticulum now is going to work alongside with the Golgi apparatus. So now, let me digress. Here, you are going to notice that the DNA that is contained in the nucleus is going to be expressed. That DNA that is expressed is going to cause the production of proteins. These proteins, many of them, are going to transit through the rough endoplasmic reticulum. The rough endoplasmic reticulum here is going to be essential for the synthesis of proteins. These proteins are going to be packaged, and a vesicle is going to bud off from the rough endoplasmic reticulum. That vesicle, that now we call a transport vesicle, is going to migrate toward the Golgi apparatus. There, the cargo is going to be released in the Golgi apparatus. The cargo is going to undergo some modifications, and this modification are going to determine the fate of these proteins. Some of the protein are going to be secreted outside of the cell here. So you have a secretory vesicle that forms, that buds off from the Golgi apparatus and going to migrate toward toward the plasma membrane, and the content is going to be secreted outside. So this is one of the things that is happening. Now, remember, I told you that you have the rough endoplasmic reticulum. That endoplasmic reticulum is rough because you have some ribosomes in yellow here, right? Some ribosomes that are attached to the plasma, to the membrane of the endoplasmic reticulum, causing biologists to see that endoplasmic reticulum to look rough under electron microscope. On the other hand, you are going to have some smooth endoplasmic reticulum right here. This smooth endoplasmic reticulum is going to be involved in the synthesis of lipids, and it has many other functions such as detoxification. It really depends on the type of cell, right? The function is going to depend on the type of cell. For instance, if you consider muscle cells, the smooth endoplasmic reticulum, it is going to be mainly for the storage of calcium, right? In liver cells, the smooth endoplasmic reticulum is going to be there for detoxification mainly, right? So you have lipids that are produced, and those lipids are going to migrate through transport vesicles, right? They're going to migrate to the Golgi apparatus and so on. So now, the proteins that are synthesized through the rough endoplasmic reticulum then migrate to the Golgi apparatus are going to be either secreted outside of the cell. Some protein may be integral part of the plasma membrane, so they are going to be embedded in the plasma membrane. We are going to see this later. Some proteins are actually enzymes that are going to end up in little sacs that we call lysosomes. These little sacs that we call lysosomes are going to be filled with digestive enzymes. These digestive enzymes are responsible for breaking down anything that the cell brings in, such as, for instance, if the cell engulfs a particle from outside, for instance, a bacterium that is roaming around, causing an infection, a virus that is moving around causing an infection, well, our cells are going to engulf them, form a vesicle. That vesicle is now going to come and merge with the lysosome to form what we call a phagolysosome. Okay? Phagolysosome. All right? And that phagolysosome is now going to be the structure in which the particle that was brought in is going to be digested. So the membrane system, the rough endoplasmic reticulum is going to have ribosomes attached to the membrane, and those ribosomes are going to be responsible for the production of proteins that we find in the rough endoplasmic reticulum. Ultimately, these proteins are going to burn off in a vesicle to migrate toward the Golgi apparatus. Then we have the smooth endoplasmic reticulum. This smooth endoplasmic reticulum is smooth because it doesn't have ribosomes attached on the surface, right? They are very much involved, the smooth endoplasmic reticulum, in making carbohydrates and lipids. And then we have the Golgi apparatus that works in close relationship with the rough endoplasmic reticulum. The Golgi apparatus, its job is to process the proteins that come from the rough endoplasmic reticulum. That's one. It's going to be also responsible for packaging these proteins after modification and send these proteins where they are intended to go, whether it is outside of the cell, as part of the plasma membrane, integral part of the plasma membrane, or to be sent as enzymes in the lysosomes. So here we have an example of how complex the endomembrane system is and how they work together to produce milk. So you have the genetic information in the nucleus that is going to be expressed, form proteins, okay? And these proteins, well, let me step back. So you have the genes. These genes are going to be converted into messenger RNA. The messenger RNA is going to be carried outside of the nucleus here, and you can see how the messenger RNA is carried out. There, the ribosomes right here are going to target the messenger RNA and start producing protein through a process that we call translation. These proteins are going to be produced through the rough endoplasmic reticulum right here. The proteins are going to be modified in there. They're going to be carried to the Golgi apparatus. So here we have the Golgi apparatus, right? The proteins, after being modified in the Golgi apparatus, they are going to burn off from the Golgi apparatus. We have a transport vesicle here that is going to cause a secretion of the proteins outside of the cell. On the other hand, though, we are going to have lipids that are going to be produced by the mammary cells. So we have the smooth endoplasmic reticulum that is going to produce lipids here. So we have the lipid droplets. These lipid droplets are going to migrate and eventually are going to be released. So here you have a fragment of mammary cells, right? That actually, when this fragment breaks, this is when we are going to have the milk coming out. Okay, so you have really here the milk production with gene expression. The messenger RNA is going to exit the nucleus through nuclear pores. The messenger RNA is going to migrate, and the ribosomes are going to cause the translation of the message in the messenger RNA into amino acids, and these amino acids eventually are going to form proteins. That's one. Two, we are going to have some lipids that are going to be produced by the smooth endoplasmic reticulum. Some sugars are also going to be produced by the smooth endoplasmic reticulum, and eventually all these components, proteins, carbohydrate, lipids, are going to be found as secretory chemicals in the milk. So lysosomes are little sacs that contain bunch of digestive enzymes, a lot of different kind of digestive enzymes, right? So lysosomes are membrane bound organelles that actually obtain enzymes from the Golgi apparatus. Okay, these enzymes are going to break down all kind of things, including bacteria, viruses, worn out organelles, and even cholesterol. Now, mind that in the worn out organelles, we are going to have what we call autophagy. This is basically when something inside of the cell is going to be targeted by our own lysosomes to destroy them. For instance, a worn out, say, mitochondrion. That worn out mitochondrion will need to be broken down and recycled, right? So beyond lysosomes, we're going to have vesicles. These are small membranous sacs that are used for many purposes, including transport. I mentioned earlier transport vesicles. So here you have an example of how some particles are going to be brought inside of the cell through a process that we call phagocytosis. So phagocytosis. Now, the vesicle that comes in, that is called a phagosome, is going to come and merge with a digestive organelle that is the lysosome. So now the lysosome is going to merge with this phagosome to form a phagolysosome that is going to digest the particle that was brought in. Now, this is in case of phagocytosis. Now, the particle that is brought in can be a bacterium, it can be a virus, but we can also have some worn out organelles such as a mitochondrion here. In the process of autophagy, right, we have that this worn out organelle is going to end up in a vesicle, and that vesicle is going to merge with a lysosome. The digestive enzyme in the lysosome is going to cause a breakdown of the particle that was now engulfed. So lysosomes are going to differ in number based on the cell type, right? Cells, for instance, of the liver are going to have a lot of lysosomes compared to muscle cells. The cells that are specialized in capturing bacteria, such as a macrophage, is going to have a lot of lysosome compared to, say, compared to a skin cell, for instance. So macrophages are going to have a lot of lysosome because they break down bacteria, viruses. Liver cells are going to break down cholesterol, toxins, and drugs, so you bet that they are going to need a bunch of lysosomes. So we also know, remember this is a human genetics class, that absence of some enzymes in the lysosome can cause major issues in people, right? We have some instances of genetic diseases where people are born with some defects, and those defects are basically centered around a missing enzyme. Now, for instance, the Tay-Sachs disease, it's a disease that is a recessive disease, inherited disease. That disease affect the central nervous system because of lack of one enzyme that is designed to break down lipids in individual with the Tay-Sachs disease. The accumulation of the lipid in this individual, and especially in the brain, is going to cause the nervous system to degenerate, right? In general, people with Tay-Sachs disease don't live long. They going to die within the first 3 years of their lives, just because of that tremendous accumulation of these lipids. And again, that accumulation is going to occur because of the lack of enzyme that are responsible for breaking down those lipids. The peroxisome are very similar to lysosomes. However, they are different in the sense that they don't contain as many digestive enzymes as lysosomes. That's one. Two, they are going to be involved very much in the breaking down of lipids in a process that we call beta oxidation. In a process that we call beta oxidation. Okay, that's a breakdown of lipids. Peroxisome also participate in the synthesis of bile salt. They play a role in detoxification. We use oxygen, and one thing about oxygen is that it can cause the formation of hydrogen peroxide. Now, we do have peroxidases, which job is going to be to convert hydrogen peroxide into water and oxygen gas. Okay, peroxisomes are the one that do this job. Peroxisomes are going to be abundant in the liver and in the kidney. Now, there are some instances when people can have a genetic disease around peroxisomes. There is a particular condition that is called adrenoleukodystrophy. That adrenoleukodystrophy, it's an example of genetic disorder where there is absence of a peroxisomal enzyme. Again, adrenoleukodystrophy, it's a condition that is caused by the absence of a peroxisomal enzyme. The mitochondria are membrane-bound organelles that have two membranes. You have an outer membrane and an inner membrane. So we have an outer membrane right here, and we have an inner membrane. Now, obviously, between the two, you have an intermembrane space. Now, mitochondria are very specialized organelle in a sense that they are responsible for producing much of the energy that we need for cellular work in our body. That energy is in the form of adenosine triphosphate, or ATP. Much of the ATP we produce is produced by mitochondria. Now, mitochondria are also very characteristic for the fact that beside the nucleus, this is the only other organelle that has some DNA, one, and ribosomes. So our mitochondria contain DNA and they contain ribosomes, which means that this DNA that can contain genes, and these genes are being expressed through transcription and translation. The ribosomes in the mitochondria are going to translate the messenger RNA from this DNA into proteins. Actually, there is a theory that mitochondria may have been some prokaryotic organisms that lived a life on their own at some point, right? We call this the endosymbiotic theory. I will not bug you with that, but just for your information, know that mitochondria resemble prokaryote in many, many ways. Now, because we are in human genetics here, let me tell you that the DNA that is found in your mitochondria in your body is exclusively from your mom. Because during fertilization, the sperm cells only brings the egg, nothing else. All the cytoplasm that is found in the zygote, including mitochondria, come from the egg, which is from your mom. And this is the reason why it is possible to absolutely tell who is the mother of a child, but it's not always absolutely possible to tell who is the father of a child. You have mitochondrial DNA in your cell that is exclusively from your mom. So this slide here, it's really to give you a quick summary of the organelle in the animal cell. So please go over that slide. You're going to have the organelle, you're going to have the structure and the function, right? For instance, the endoplasmic reticulum. The endoplasmic reticulum, it's a membrane network that contains the rough endoplasmic reticulum that has ribosomes attached and the smooth endoplasmic reticulum that does not have ribosomes attached. Now, what is the function? They are going to be responsible for protein synthesis because of the ribosomes that are attached. They are going to be important for the folding of proteins, lipid production, and carbohydrate. It's not mentioned here, but carbohydrate are also synthesized through the endoplasmic reticulum. So you're going to do the same thing for all the rest of the organelles, including the Golgi apparatus, lysosome, mitochondria, nucleus, peroxisomes, ribosomes, and vesicles. Now, let's take a look at the membrane, the plasma membrane. The plasma membrane is made up of a lot of lipids, a structure that is mainly made up of phospholipids. The phospholipids are characterized by the fact that they have a head that is hydrophilic and a tail right here that is hydrophobic. Now, in a water-based environment such as the body, these phospholipids, when they are in large amount, they will spontaneously form a bilayer. So this is one layer and this is another layer, so that we call the plasma membrane a phospholipid bilayer. So phospholipid bilayer. Okay, now that phospholipid bilayer is going to be selectively permeable. It allows only some chemicals to get in or out, while others will never be able to get in or out on their own. In addition to phospholipid as part of the plasma membrane, we are going to have cholesterol. In human, we have a bunch of cholesterol, and that cholesterol plays a very important role in the fluidity of the plasma membrane. In addition to cholesterol, we are going to have proteins. These proteins that are embedded in the plasma membrane are going to play different roles. Some of them are going to play the role of channels through which chemicals can go in and out. Some of them are going to be able to sense what is going on outside. Some of them are actually going to be surface markers to tell one cell apart from another type of cell. For instance, a red blood cell is going to be different from a white blood cells. Now, in the body, how do cells tell each other apart? They are going to tell each other apart because of all the surface markers right here, right? When a protein has some sugars attached, like here in green, that protein is called a glycoprotein. When sugars are attached to a lipid, we have a glycolipid. Okay? In green, these are carbohydrates. Okay? Now, you're going to notice that carbohydrates are highly abundant on the cell surface toward the outside of the cell. But toward the inside of the cell, you are going to have many protein fibers here on which some proteins are going to be attached. These are the microfilament fibers that are part of the cytoskeleton of the cell. So the membranes that are embedded in the plasma membrane are going to play a very important role in creating a specific identity for a particular type of cell. Some are going to be essential for the transport of molecules. They're going to be essential to keep out toxins and pathogens. Some are going to function as ion channels, some as transporters, some are going to be important in cell-to-cell communication, and then used as receptors. So they're going to be point of attachment of signaling molecules. Some can be essential in cellular junction, such as in epithelial tissues, where two cells are going to come very closely attached together via what we call tight junctions. Okay? Now, needless to say that this protein can also be the reason for some diseases when these proteins are defective. Remember, in chapter 1, we talked about cystic fibrosis, right? Cystic fibrosis is a type of channelopathy. A channelopathy is basically when a channel is faulty in a person, right? And we did see in the previous chapter that the CFTR proteins, when they are mutated, they are not able to allow chloride ion in and out of the cell properly, so that you have an accumulation of mucus outside of the cell. Going further, we have the cytoskeleton. The cytoskeleton, it's basically the skeleton of the cell. That skeleton of the cell is going to be made up of three types of fibers. The first type of fibers we're going to address is going to be the microtubules. The microtubules are the thickest cytoskeletal fibers found in the body. These microtubules are made up of proteins called tubulins. We have two types of tubulins. We have the alpha tubulins and the beta tubulins. Okay, and these tubulins are going to come together. So this is the alpha, this is the beta. They're going to come together to form dimers. These dimers are going to end up forming structures like what you see here, right? And this structure actually looks like a straw under a microscope. These microtubules are very characteristic by the fact that they are very stiff, okay, and they are thick. The second type of cytoskeletal filament we are going to address, it's the microfilament. The microfilament are also called actin filaments. These actin filaments are called so because they are made up of proteins called actin proteins. These actin proteins form thin filaments. Here you can see actin filaments right here attached to each other, forming long structures. These are flexible and contractile. They play a very important role in the movement in the body. And then the third type of filament that is part of the cytoskeleton is going to be the intermediate filament. We call them intermediate filament because of the intermediate size between the microfilament that are thin and the microtubules that are large. So those ones are going to be intermediate, are going to have an intermediate size, right? So the cilia and flagella, which are structures that are appendicular to the cell, are going to be essential for motility, for motion. In the human body, only one type of cell have a flagellum. These are the sperm cells. The flagellum allows the cell to be able to swim. And that flagellum contains some microtubules. Also, the cilia are going to contain microtubules. The cilia are going to exhibit a wave-like motion. That wave-like motion is going to allow that structure, the cilia, to push mucus up and down. For instance, if you consider your throat, the surface of your windpipe is lined with